Up next, we explore groundbreaking research into how countless bacteria in our bodies influence our health and well-being. UNC Charlotte professor of bioinformatics Anthony Fodor played a critical role in the National Science Foundation's Human Microbiome project. Inside UNC Charlotte's Lisa Patterson caught up with Dr. Fodor to discuss the results of the project and his latest research collaboration.
Each person is an ecosystem. We know that our individual ecosystems are comprised of genetic material passed to us from our biological parents. We've only recently learned that our ecosystems include a whole host of microbial communities coexisting within our bodies.
There are something like 10 times more microbes within the human body than there are human cells. And there are encoded within the genes of those microbes are something like 100 times more microbial genes than are encoded in the human genome.
UNC Charlotte professor of bioinformatics Anthony Fodor played an important role in the multi-year NIH-sponsored Human Microbiome project. The project mapped the distribution of microbes taken from 18 sites on the bodies of 200 healthy people with the goal of understanding how different microbes contribute to health and disease in the human body.
The Genome Project, more than 10 years ago now, was designed to kind of ask the question, what is the structure of the human genome? And the Human Microbiome Project, which has just been completed, is designed to ask the question, what is the nature of the human microbiome?
A healthy adult human harbors some 100 trillion bacteria in his gut alone. The microbiome, as the body's bacteria are collectively known, includes around 3 million genes. Prior to joining the Human Microbiome Project, Dr. Fodor's lab investigated how nutrition, genetics, and gut bacteria work together to affect an individual's susceptibility to fatty liver disease. UNC Charlotte alumna Melanie Spencer was part of Fodor's research team.
Each person has their own microbiome. It's like a fingerprint. And understanding the microbiome, which bacteria are most important for nutrition, which bacteria may affect chronic disease, gives us an entirely different view on things that we can affect when we're looking at treatments, but also things that help us understand diagnoses much better.
If we have a better understanding of how microbes are contributing to health and disease phenotypes, then by monitoring those microbes using this new sequencing technology, which is only going to get cheaper in the future, we might be able to say, you know, you're actually at risk for this disease, and we know this because you have microbial dysbiosis, and we really couldn't have known that any other way.
Dr. Fodor and his team are working with collaborators at other universities to examine inflammatory diseases of the gut and their relationship to colorectal cancer. One collaboration yielded results that were published in the August issue of the journal Science.
We knew that microbes have an impact on cancer. We have published in 2009 a paper showing that different microorganisms were leading to a different extent of cancer. So we designed an experiment using germ-free mice which don't have any microorganism in them, and we introduced different bacteria, and we found out that the potential to induce cancer was related to how much inflammation the mouse was developing.
Christian has actually found that you can get mice to progress to cancer if you put some kinds of microbes in the mice but not others. And so what that suggests is that there's some interaction between our immune system and the microbes that in some cases, in mice that are predisposed to have inflammation anyway, can damage cells and potentially progress them to cancer.
At UNC Charlotte, faculty and students in the Department of Bioinformatics and Genomics utilize cutting-edge technology to process enormous amounts of data from large-scale experiments.
Bioinformatics is the marriage of biology and computer science. As we've gone into genetics and genomics, we've realized that there is so much information packed into DNA, and we need very good computational techniques to mine that information.
We sequence DNA and we generate terabytes of data. So we need bioinformatics to crunch numbers and come up with identity of microbes we're studying.
It's not really that computing has changed that. The theory hasn't changed. It's just what we can do with it has changed because it's so much more mobile. And so we're at the very beginning in some ways of biotechnology because it's something that gets done in labs like this one.
Advances in technology have made initiatives such as the Human Microbiome Project possible.
We couldn't have done the project if we didn't have really big hard drives. If you go back 10 or 15 years, you couldn't buy a 2 terabyte hard drive. Now you can buy one on eBay or at Newegg for $125. And we filled up many of these hard drives with data. The basic foundation has to be there. And in this case, we need fast computers, we need lots of cheap processors, and we need lots of cheap hard disk space.
While great strides have been made in identifying the elements of the microbiome, researchers are just beginning to unravel the microbiome's contribution to chronic disease and many other disorders.
You might make a prediction. Well, if this bacteria has these 14 genes, and if I just take out this third gene, or if I find something that will inhibit that third gene, the bacteria won't be able to survive. But often the bacteria has ways it'll find another way, right? There's some pathway that's not the pathway that you're looking at. So we're still learning how to read a genome sequence, I would say. That's one of the great challenges of my field, is how do you parse this information? But again, these are early days. I think it may take us 100 years, right? But from a historical point of view, 100 years isn't that long in science. We've been doing science for hundreds of years.